And thanks everyone for taking time to tune in today. And I encourage you to check out the rest of these lectures. They're always very interesting. So I'm going to start. As Boris mentioned, I did spend a lot of my career thinking about above ground litter, really, and its decomposition, like many foresters or ecologists who started their careers in the previous century. This was what we thought was going to lead us to a better understanding of soil organic matter generation and nutrient cycling. So this was a major theme of my research from the time I put in my first litter bags in 1982 under a meter of snow to the when Lars and I synthesized pretty much what we know in this discipline some 40 years later in this paper. And during this time, Doug and others made it clear that we need to think about below ground litter as well, both roots and mycorrhizal fungi. And so a lot of people have done that. And I think we have a pretty good handle on that aspect of soil organic matter generation now. But for the last 20 years, I've been thinking in the back of my mind and then in the front of my mind, if there isn't another important carbon flux in forests that we need to think a lot more and find out a lot more about. And this started with this paper in Nature, which many of you will remember, the Heubergs paper from their girdling experiment in a pine forest in boreal Sweden, which showed that half of the biological activity in the boreal forest soil was fueled by carbon that was fixed through photosynthesis a few days earlier. And this was kind of disturbing at the time because it suggested there's a lot going on other than litter and its decomposition. And there's a lot of carbon coming down and going out into the soil. And this is being shown now in more detailed studies. Um, this paper came out of the Swiss canopy crane experiment, and they were able to label, they labeled canopy, and then they uh, moved litter from one plot to another, transferred litter. And so they were able to have either litter labeled or roots labeled. And then they sampled the below ground arthropods, and they found that just about all of them obtain their carbon from the soil and not from roots, and not from above ground litter. And then the Heubergs followed their work up with some carbon labeling experiments, and they found that labeling the leaves and so the photosynthate, they were able to trace it and found it after 24 hours already down in the stem through moving through the phloem. And then after 2 to 4 days, it was in soil respiration and it was in the free living microbes in the soil already 2 to 4 days. And another experiment, they found the label in ectomycorrhizal fungi, not too surprising, within 2 to 4 days, but then in the collembola that graze on these, suggesting that they're probably actually taking some very recent photosynthate out of these ectomycorrhizal fungi. And then Sue Grayston and Carolyn Churchland and Maria Briones did an experiment in England. They labeled Sitka spruce, they stem injected it with labeled glucose, and they found the label in the soil DOC 2 to 6 days after. And then within 2 to 4 days or 4 to 6 days, they found it in the soil microbes and then in mites and enchytraeids as well. And so these, and this really cool experiment from Christina Kaiser's group showed that labeled photosynthates from plants were not only transferred to ectomycorrhizal hyphae within 24 hours, they were out of the ectomycorrhizae and into the bacteria that line their mycelia. So this means that they're not only going into the mycorrhizal fungi, but they're also being exuded from those hyphae and being taken up by bacteria, which then, of course, can become part of the food chain. And interestingly, they found that when they added nitrogen to one of the litter compartments, this incorporation was very strongly reduced. And I'll ask you to keep that in the back of your mind. And then we have this paper from Seattle where they provided an estimate of kind of the average amount of hyphosphere that we would have in a forest soil. And you can see, if you think not just roots exuding label or photosynthate, but also hyphae, there's an average of 2,000 centimeters of hyphae in a cubic centimeter of soil. So there's a huge surface of mycorrhizal hyphae that is available for exuding and transferring carbon into the soil biota. And of course, now we know that also these microbes, bacteria, they also release all sorts of carbon compounds, whether it's biofilm or extracellular polymeric substances. There's vast amounts of this being exuded. And this, of course, is coming mostly from the plants through the mycorrhizal fungi or the root exudates and into the bacteria. So there seems to be, I think, a lot of carbon that's coming into the soil and the soil food web from this conduit. And so I think that these fluxes of labile carbon from living tree roots and associated mycorrhizal fungi might be really important sources of food and fuel for the below ground food web. And of course, this would get into the food web through a number of pathways. On the right, you can see what I just described. It's exuded out of roots or mycorrhizal hyphae, taken up by microbes, and then these are grazed by protozoa. These all turn over or are grazed by larger organisms and so enter the soil food web. Or they can be grazed, the fungi can be grazed by collembola, which again largely take that labile carbon out, and then they're consumed by other things, or even just root feeding nematodes, sucking it right out of the roots and then entering the food chain. And then over the last 20 years, it's become apparent that contrary to our thinking before that most soil organic matter came from litter, above ground or below ground, it seems that most of it has been processed by microbes. And if that's a major source of soil organic matter, then you have to think that since a lot of microbes prefer labile carbon, that as Catrufo proposed, this could be labile plant constituents could actually be a dominant source of these microbial products that become soil organic matter. And then, of course, Sokol found that inputs from living roots are very efficient in creating both slow cycling mineral-associated organic matter as well as particulate organic matter. So this suggests to me that these fluxes of labile carbon from living tree roots and mycorrhizal would also be really important for replenishing the soil food web. And of course, replenishing soil organic matter. And of course, because soil organic matter is constantly being degraded, as it has to be constantly replenished, or else the amount of it will decline. So that led me to start thinking a lot about, in the front of my mind for a change, about why trees and their associated mycorrhizal fungi are exuding fixed carbon below ground. And there's a lot of hypotheses about these, or theories, or things people say. But my null hypothesis for this had always been that plant exudates are mostly either metabolic waste or surplus metabolites that plants produce that they have to get rid of somehow. And so this is a really fun way to get soil microbial ecologists excited, is to suggest this. So I would just suggest it and not push it any farther. But about 5 years ago, I thought it was time to look into this because I hadn't seen anything that had convinced me otherwise, despite all of the the rather intriguing ideas out there about why they do that. And so what I discovered was this fact that plants often have surplus carbon. I didn't discover this, I just found out, and we've known this forever. So I don't know why we don't consider this more and why we consider to think about carbon as a really precious substrate that plants have to be very efficient and almost thoughtful in how they spend it. So here's some examples I found. Harper's textbook, he said the plant may indeed be a pathological overproducer of carbohydrates. And then there's this delightful paper I encourage you to read, even if just for fun. The capture and gratuitous disposal of resources by plants, and they're talking about fixed carbon mostly. And then, of course, Christian Corner has been pointing this out for so long now that plant carbon reserves are rarely even close to depletion. And then Pete Millard reviewed all the evidence and concluded that the growth of trees is not carbon limited. So, and I recently found out this from a colleague who works in marine ecosystems that they've known forever that algae, from phytoplankton to kelp, they exude carbohydrates into the water all the time. This ranges from 5% when they're in nutrient-rich waters to 40% when they're in nutrient-poor waters. And there's that nutrient link again. And they consider this, or at least did at that time, to be probably an overflow mechanism when photosynthesis takes place more rapidly than is required to supply the needs of growth. So very familiar. So this has been known for so long. So why do they produce surplus carbon? And here I'm not talking about an evolutionary reason. I'm just saying it's known that they produce surplus carbohydrates because photosynthesis and leaf growth are both limited by the availability of nutrients and water, but they have different sensitivities. They're very distinct in their sensitivities. And it's known that growth is curtailed at an earlier or lesser stage of deficiency than is photosynthesis. And this is nicely laid out in this paper I encourage you to have a look at by Müller. And what you can see is as water stress increases, or water deficit increases, you can see, I've drawn it out for you, make it a little clearer, growth plummets at quite an early stage of water deficit, whereas photosynthesis continues. And then only when the deficit starts becoming severe does photosynthesis decline. So there's this area in between, which I'm calling an intermediate deficiency, where growth has stopped, but the plant is continuing to photosynthesize. So after much research and painful reading of plant physiology books and discussion with some very knowledgeable colleagues, we put together this hypothesis, which was published in Tree, that under common environmental conditions, plant leaf cells produce more photoassimilates than they're able to use for primary metabolism and growth, and so they have surplus carbon. And you can see along the top in the yellow, the conditions which give rise to this are either limitations of nitrogen, phosphorus, or water, or high CO2 concentrations, or actually high light as well, or cold temperatures. So leaf growth decline, carbon fixation continues, surplus photosynthates are produced, and these surplus carbohydrates can be transformed to other compounds. So the plant will put them into whatever it can that does not contain the limiting nutrient, and so it's able to recycle that nutrient and keep photosynthesizing. So if nitrogen's limiting, it puts it in starch or various secondary metabolites that don't contain nitrogen. Or if phosphorus is limiting, put it into amino acids and organic acids. And the other thing they can do is just remove them from the leaf because these deficiencies don't interfere with phloem loading. So they stuff them in the phloem and they go through the stem, and some will end up down in the roots where they could be used either for growth if conditions are suitable, or can be stored as starch, or they can be transferred to mycorrhizal fungi, or they can be exuded, or they can be exuded from mycorrhizal fungi, and then they enter the rhizosphere and hyphosphere. And from there, they can go through the soil food web and can also eventually be turned into soil organic matter. And so when viewed through this lens of plants producing surplus carbohydrates under certain conditions, I found that a lot of kind of phenomena that happen in forests that had popped up, into my realization during my career, they're actually quite predictable if you look at them through the lens of surplus carbon. And this includes several below ground phenomena. Of course, this is when things always get interesting, is when we go below ground. This includes nitrogen fixation, and I've been talking with some colleagues who study this because they're wondering why nitrogen fixation continues at pace even when there's loads of nitrogen in the environment, if there are also drought conditions. And this has been kind of a curious thing they've been wondering about, but it's perfectly consistent with surplus carbon being produced under those conditions and going to the roots, and they're getting them. And I'm not going to go into the others, but I am going to encourage you to have a look at this paper, which just came out in New Phytologist, where a bunch of really great scientists accepted the challenge to look into this question. If mycorrhizal fungi only receive surplus carbon from the plant, do we need to invoke trading or sharing to explain the transfer of carbon from plant to fungus? So, and they did a very careful and thorough analysis of this question. So I encourage you to have a look at that. And all these papers are open access, of course. But I want to move on to the management implications, which I began thinking of after working on this hypothesis and becoming more and more convinced of this this carbon flux, this flux of labile carbon in forests. So the question is, how can we manage forests to maintain or promote this flux of labile carbon from trees to the below ground ecosystem? And the first thing I thought of is evident in that photo taken only half a kilometer from my home. And I think we can be forgiven when we thought that most soil organic matter and fuel for the below ground ecosystem came from above ground litter. I think we can be forgiven for thinking that it was probably all right to clear cut forests as far as the below ground ecosystem is concerned, especially when you know that a third or about a third of the biomass that is generated from harvesting forests, I should say necromass, is actually below ground. So there's all of this, plus there's all the dead roots and mycorrhizal fungi for animals to feed on and to generate soil organic matter eventually and soil carbon. So I think we can be forgiven for thinking that, but now I look at this quite differently when I think about this flux from living roots of trees, from roots of living trees. And so one suggestion, the most obvious one I think, is that we should be maintaining some living roots if we want to sustain the below ground ecosystem and replenish soil carbon, even while harvesting forests. And of course, this cries out for retention forestry or continuous cover forestry, as you more commonly call it here, and various other sorts of forestry that just say don't remove all the trees. And so we propose that we occasionally refer to this as continuous root forestry, um, because a lot of the benefits of continuous cover forestry may be actually below ground benefits. And it would actually be a good thing to make people think about the below ground ecosystem once in a while. So then the question is, well, how many trees do we need to retain? So we did a thorough search of the literature where people had actually laboriously quantified roots and mycorrhizal fungi with distance from tree stems, as well as some carbon isotope signature of soil microbes with distance from the forest edge. And fortunately, these both led to the same sort of conclusion, that the influence of living tree roots below ground is gone by about 10 meters, and it's severely reduced by 5 meters. So this leads to the suggestion that the way that we can continue to maintain this flux of carbon throughout that cutover is to minimize the area of the cutover that's more than 10 meters from a living tree stem. And here's an example from Vancouver Island. You can see this is a dispersed retention treatment, we call it, about 40 trees per hectare, which would be about 15 meters stem to stem. And of course, clumps of trees are even better, but they have to be distributed through the cut block and not like this, which is so common in British Columbia now. Clear cut with reserves, it's called. This will not serve the function of promoting life below ground. So another way I think we could maintain this flux of carbon in managed forests is doing what I call ecologically intelligent nutrient management, which is somewhat different from economically intelligent nutrient management. And so that's mostly through optimal fertilization, which I'll explain, as well as use of nitrogen fixing species. So we know when we, especially when we add nitrogen to a nitrogen limited forest, what we always find is increased foliar nitrogen concentrations, greater rates of photosynthesis, more leaves, and greater above ground biomass, including the stems, of course, which is what we're interested in. But below ground, it's very different, and we often find reduced fine root biomass, reduced mycorrhizal abundance. You can see this Tressiders meta-analysis shows less mycorrhizae when you add nitrogen or phosphorus, and interestingly more when you elevate CO2. And then more studies from Sweden showing reduced respiration, reduced microbial biomass, and reduced fungi when you add nitrogen to boreal forests. And this was demonstrated that this is related to the decline in below ground carbon flux following the addition of nitrogen to these forests. So they found a year after nitrogen addition, the flux of tree photosynthate to the soil biota was reduced by 60%. And they were also able to track the recovery of ectomycorrhizal fungi when they stopped fertilizing a previously nitrogen saturated forest. So all this suggests that this is related to nitrogen fertilization, reducing this flux of carbon below ground, and so having all these reductions in a lot of the activity normally going on below ground. So this prompted me to suggest that maybe what we should be doing is dialing down the amount of fertilizer we add, which we usually add to maximize above ground growth. And maybe find the optimum point where you can have a nice increase in above ground growth and good growth rates and timber production and all that, but still maintain a flux of carbon to support the below ground community. And this will also enable it to not have a reduction in fine roots and mycorrhizal fungi, which of course is so important if there's a chance of the forests encountering drought. So it should make them more resilient, as well as promoting soil life, as well as soil organic matter generation. And the other part, of course, is using nitrogen fixing tree species, which we don't do nearly enough. It's just always so easy to just add nitrogen fertilizer with all its shortcomings. And of course, Dan Binkley, Dick Fisher put this together, showing that you can get some very substantial inputs of nitrogen, upwards of even more than 100 kilograms of nitrogen per hectare annually by having alder, either pure alder or mixed alder stands. And increases in the basal area of crop trees has been demonstrated. We've also shown, it's been shown worldwide, and we found it in this review that Matthias Meyer was author of. Consistently greater soil carbon stocks under stands that include nitrogen-fixing tree species globally and consistently. And then there's also been evidence that there is more soil carbon in stable fractions under nitrogen-fixing tree species. And lots of other good things about nitrogen-fixing species. They grow really quickly. Studies in the Northwest showed even just 5 years of an alder stand was enough to significantly increase soil organic matter and nitrogen. It builds soil carbon stocks faster than other species. I think a lot of these effects are because nitrogen fixing plants exude nitrogen in various forms as well as carbon, which is really what you need to build soil microbes. And they have distinct soil microbial communities. So they're especially important, I think, on degraded soils. And they're also best in mixture and not in pure stands. Too much of a good thing. And the last way I think that we could promote this below ground flux of carbon and what I'm calling regenerative forestry, because that is really forestry that's focused on promoting soil life, is encouraging plant diversity. And some of the best evidence for the values of plant diversity below ground have come from the YINA experiment, so not yet in forests. So here they found that higher plant diversity increased rhizosphere carbon inputs, and this increased microbial activity and carbon storage. And they found that it's not just enough to have different species. The biggest results come when you have plant species that are distinct, either phylogenetically or especially functionally. And I think this is related to the nature, them having different metabolisms and different exudate profiles. That's a hypothesis, but it's consistent with this interesting paper, also from Jena, where they found a link between plant diversity and soil microbial diversity. And they demonstrated that this is probably related to the exudates because they collected the exudates from all the plots of various diversity and made cocktails of those, and then applied those cocktails to soil, to a kind of a control soil, and they found the same results just by using these exudate cocktails as they found in these plots. That high plant diversity generates a lot of diversity of exudates, and this generates a lot of diversity of soil microbes. So thinking about this in forests and thinking about the way we tend to simplify our forests, makes me think we need to have more species, especially different functional groups, which brings us back to broadleaves. In systems, well, certainly in Canada, where we're so focused on conifers as our crop trees, these tend to be removed or discouraged. And yet soil broadleaves have been known for so long to be soil improvers. They change the soil biota. They create more of a mullish sort of humus. They increase the organic matter content of the mineral soil. And this was interesting, a link between, they tend to have higher calcium, and there's an interesting link between calcium and generation of microbial biomass. And then calcium also encourages the association of this carbon with soil minerals. And so what you end up with is more soil organic matter, including more stable soil organic matter. I've explained this poorly, but read Shabtai's paper. And then, of course, with the calcium and other aspects of earthworms, you tend to get broadleaves, you tend to get more earthworms, and that gives you more aggregates, more necromass, and more stabilized soil carbon. So the final thing that I'm suggesting would probably help with this aspect of forests is actually encouraging forests with more than a single species or functional group. And so I'm just going to conclude about that, what I'm calling the principles of regenerative forestry that's focused on promoting soil life and function. And so take some stems, don't take them all, and leave enough trees to keep the soil alive while the next generation of trees becomes established. Add nutrients where and when they improve the soil and foster soil life, and not just feed the trees, often at the expense of soil life. And to aim for that sweet spot of nutrient supply where both above and below ground productivity are optimized. And manage forests with more than one tree species. Embrace mixtures with broadleaves and nitrogen-fixing species, especially. Have a fallow at the start of the rotation, or grow them as mixtures, or simply don't remove them, as we still do. And finally, we need to consider the below ground ecosystem when managing forests for resilience. And I think I'm preaching to the converted here, but we really need to get this message out. We have to start thinking more about the below ground ecosystem now that we know so much more about it than we did 20 or 40 years ago. So regarding this seminar series, Challenges in Forest Ecology, I'd like to put a challenge out there for you. So everything I've been talking about can be considered hypotheses. That's based on just thinking about forests for 40 years and then a really deep dive into this topic for the last 5 years. But it's still hypotheses, and it really needs testing. So please pick your favorite and test it. And I'd like to invite you to look at these papers in which we provide a lot of the evidence that I've kind of skimmed over today, including this last one that just, as I said, just came out last week. And thanks very much for your attention, and I look forward to discussion.
Yeah, thank you very much, Cindy. This was really, some provocative suggestions, I think, for some, some comforting thoughts for others. I think it was a very good overview. So there are not so many questions yet. I will come to them soon in the questions. So please, the audience, start now to type in your questions to the chat box. But maybe I take the opportunity to start with one, because you talked about continuous root forestry, and Michael Scherer-Lorentzen, he has something similar. So in your opinion now, from your diving into the topic the last 5 years, with what plant functional types would serve for this continuous forestry? You think about shrub species, grasses immediately growing. Is it about which kind of information we know we need to say they they benefit below 1 carbon? Is it the type of exudates? Is it the amount of exudates? Or where are they exudated in the soil profile? It'd be lovely to have your opinion on that.
Yeah, yeah, you're quite right. All of those things would matter. And uh, it seems to be diversity that matters. And I think something we haven't thought enough about is how plants of different species have very different metabolisms. And as ecologists, I mean, I certainly steered away from that because I mean, it's having so much biochemistry and all that. Um, but I think we tend to not recognize the diversity of compounds that are both in the litter, in the leaves and the litter and all the other tissue, but especially in those exudates, and how this changes according to the not only among the tree species, but according to the conditions it's growing in, the soil type, possibly the climate, and also phenologically with the time of year. So a first start would just be to say, have lots of species in there and try to manage it in a way that encourages a diversity of plant species. But at the same time, we really need to know more about uh, the differences or similarities among tree species in the, the nature of their exudates. And this includes the exudates associated with their mycorrhizal fungi. And this could be profoundly different if you have different types of mycorrhizal fungi in the system, if you have AMs, as well as ECMs or ericoids, or all of them in some systems perhaps. But also the species of fungus could be quite distinct in their exudation. And we know so little about this. I was introduced to this around 1990. There was a paper Sue Grayson wrote about, and it just had a table, and it had all of the organic compounds that had already at that time been found to be being exuded from tree roots of various species. And it was just astonishing. And at the time, I remember just sitting there thinking, why are they doing this? Why are they exuding all this stuff out of their roots? So we know that they are, and we know that they're distinct, and we know that they have that distinction creates different soil microbial. There's just a universe of things going on down there that we need to know more about.
Very encouraging for the younger scientists in the audience that there is enough to do in the next 40 years.
Oh,
And
goodness,
so
yes.
let's turn our attention a bit to the chat. There is a question from Giorgio Macciano. I don't know if I pronounce it. And he asks, do you think that actions 1, 2, and 3 could be translated in quantitative estimates of additional carbon relative to BAU forestry and hence generate offset carbon credits? Unfortunately, I don't know what is BAU forestry, but maybe you know.
Yes, and I've been pressed for this on occasion, and I have to say, no, uh, we don't know, and I'm hesitant at this point to make a suggestion. So another huge research question is how does all this relate into carbon stocks? Everybody wants numbers for that, and we don't have them for this flux.
Mhm. I would like again to encourage everybody to post potential questions to the chat. But we have another one from Dan Binkley. He asked, on nitrogen poor soils, retaining large trees inhibits establishment and growth of regenerating trees. If many trees are retained, is it an issue that new trees do not do so well?
Yeah, um, it is an issue, and we have, of course, had this discussion a few times. And my response to that is yes, this will, in most systems, trees close to an existing tree will not grow as fast as those distant from it. So that the trees are still competing with one another. And the big trees will be casting shade, they'll be taking water, they'll be taking nutrients. So they could well impede the growth of the smaller trees. So it's finding that balance where you're going to get adequate growth of young trees. But what we have to consider is the ecological benefits that come with sustaining the soil life and sustaining soil organic matter generation and replenishing the below ground carbon stocks. And we have to trade that off against any reduction that we will get in the growth, above ground growth rate of tree seedlings. And we also have to consider that above ground growth rate maybe isn't everything that matters in our forests. And we're so used to thinking and considering that to be the only parameter that matters, that I think we have to change that, because our forests are facing a lot of challenging situations now, and drought is a big one amongst them. And so things that maximize above ground tree growth, if they come at the expense of either the root systems, the mycorrhizal systems, or the activity and organic matter in that soil, then I think in the long run, we may not be doing the right thing in terms of the long-term fertility and productivity of that site. So I would suggest that we need to replace the laser focus on seedling sapling regeneration rates and stand growth rates with a consideration of the broader benefits and the good that we can do, rather than just trying to minimize damage when we're managing forests.
Yeah, thank you very much. Friederike Bayer from the University in Freiburg asked what you would recommend for afforestation projects. So she says most of them are happening on former agricultural lands, which are very high in nutrient contents. And if it's a good idea to not only establish species-rich, diverse, functionally diverse forests, but also help them to get started with mycorrhiza, for example.
Um, Interesting. Yeah, not a situation I deal with here in Canada, of course. But if you're starting, yes, so you've got kind of overly fertile soil. You probably got pretty high phosphorus in there, which is going to stick around. And so under those conditions, you probably get pretty good above ground growth, but not very good development below ground. So how could you counteract that? And that's interesting, because even if you add the mycorrhizae, if the plant isn't generating surplus carbon, then I think those mycorrhizae won't be fed, and so they won't develop very well. So
Mm.
you've got to somehow get that into the situation where uh, the plant is, where it's not putting all those nutrients into above ground growth. Something's got to become, maybe it just takes time, like in that experiment of the Heubergs and the nitrogen saturated forest. If you just stopped adding nitrogen, eventually you could see the recovery. So it might
Maybe
just take.
some years of high rotation forestry to remove some nutrients to get the levels down. Maybe it's a good idea to
Yeah,
reduce it to normal levels.
yeah, but again, yeah,
Yeah.
yeah, you might as well take advantage of having that high fertility. But again, you don't want to have that system simplified in another way. It's already been simplified by having high input agriculture on it. You don't want it to be simplified by having only one tree species on it. So I wouldn't encourage uh, monocropping the forestry either.
Mhm.
But don't fertilize it anymore, at least.
Yeah, but the nitrogen deposition is so high, basically, we barely fertilize anyhow. But Douglas asked, it sounds convincing that carbon is excess, but then how are effects of elevated CO2 on mycorrhizal ectomycorrhizas explained, but also the decline after nitrogen addition?
Yeah,
Mm.
those are both actually very consistent with the generation of surplus carbon. Surplus CO2, or sorry, elevated CO2,
Hmm.
if it's sustained, then it does promote the generation of surplus photosynthates, which go below ground. And the evidence that I've seen, looked into this pretty carefully, is that a lot of that is just blasted off. Um, I forget what it was called, almost a disposal pathway. It ends up with greater respiration, and so it doesn't necessarily promote anything. Uh, promote it can promote more mycorrhizal fungi, but only if they have the other materials that they need to generate. But there's pathways like the alternative oxidase pathway, which exists in roots as well, as which seems to only exist to blast off surplus um, photosynthate as surplus carbohydrates as CO2. So that's what happens in a lot of cases with continuous elevated CO2. I forget the name of that paper, but I think it was from one of the European experiments. And with nitrogen, with reducing nitrogen to generate surplus carbon, was that the question? And then that will generate surplus photosynthates, which will then either go into secondary metabolites or be sent below ground.
Yeah, obviously, this whole your introduction triggered a lot of questions, a lot of the colleagues, because there are many questions dealing now with the question how it can be related to evolutionary theory that wasteful species are outcompeted in the end, and if this carbon-based exudates increase not the nutrient competition between plants and microbes, for example. Yeah, so
Yeah,
what kind? Yeah.
excellent question, and uh, not a new one, I admit. And so long ago, in my reading for my comprehensive exam, I read Stephen Jay Gould paper on the spandrels of San Marcos, a critique of the adaptationist paradigm. And if you read that and you think, why is it that we assume that everything we measure that any organism does in an ecosystem is adaptive? It might just not be maladaptive or significantly enough maladaptive to cause evolution to act against it. So when I go back and I see these algae, phytoplankton, like every photosynthetic organism having this funny little quirk that growth stops at an earlier stage of deficiency than does photosynthesis, we would think, no way, absolutely no way would that happen because that would be evolutionarily stupid. Can't be so, but it's so, right? And it's just so. And it obviously isn't a big problem because no plant has come up with a better solution to it. So yeah, photosynthesis, or this link between photosynthesis and growth, is not tightly coupled. And if we were designing an organism, we would make them tightly coupled, and we wouldn't allow for this inefficiency because we hate inefficiency. We do not think plants would be inefficient. But if they usually have surplus photosynthate, what's the ad advantage of being very conservative with it? If it's in surplus, there's no advantage to it. So they got this funny little quirk that growth turns off, photosynthesis carries on. They've got these places they stuff the stuff, and none of them are sufficiently maladaptive to have this end, or even to have a workaround. So most people would say, well, then it must be adaptive. Well, maybe it just isn't maladaptive. And maybe there are some things that happen because of it that
Or
work out really
maybe
well.
they're adaptive in ways we don't know yet. Maybe a little
Yeah,
bit. Maybe it's not very effective, but if you have too much of it anyhow, so doesn't
yeah.
hurt too.
So it doesn't have to be adaptive, but it can lead to some good things. It obviously doesn't lead to to really bad things, or it wouldn't exist. But this funny quirk exists, and it has all of these um, outcomes, and nobody's come up with a better way. So I would say, let's think carefully about why an organism has to be efficient with resources it has in excess.
So this maybe we take one last question because soon it's already 5 o'clock here. So from then Binkley again, if trees might generate excess, the waste carbon that is disposed below ground, might below ground ecosystems also have excess carbon to burn off? How would we know there is a good amount of carbon flowing below ground with this excess?
Yeah, well, what's excess? I've avoided the use excess and waste because
Mhm.
that seemed a bit too much, um, especially for the physiologist to embrace. So I go back to surplus. Um, and yeah, it does seem like there is a lot of this stuff that flows through the ecosystem and flows through the organisms. And I think that's why we have ecosystems like this below ground, is largely because of this flux as well as other ones that increase the diversity. But if we didn't have this, if this didn't exist, I think our ecosystems would be quite different. And this includes marine aquatic ones, anywhere there's photosynthesizing organisms. I think a lot of things have developed in ecosystems because of this flux. And and I like to help people think about this by saying, imagine in the beginning, there's surplus carbon. So as soon as there's photosynthesis, there's surplus carbon, and everything follows from that. So if you have land plants, they're oozing some of this stuff out of their roots. Why wouldn't fungi be attracted to it? Why wouldn't all these things happen? Why wouldn't bacteria be there? All these things they would just happen, and we wouldn't have to come up with all sorts of reasons why all these things are happening and why this organism is being attracted and not that organ and all of that. It would just happen.
Yeah, I really have to say thank you. I think this was a mind-provoking talk, and you see the waves are going high in the chat if you're right or wrong or it's somewhere in between. So, but it was excellent, just to put the system upside down and consider it from a different perspective. So, thank you very much again for coming here to the forest ecology really challenges in forest ecology seminar series, and also thank you for all the listeners outside
Yes,
for
thank you for coming,
tuning in.
everyone.
Thank you very much.